everyone. Thank you all for being here to our weekly research bites that we're having in place of our INKY 2020 meeting due to the whole COVID situation. We're really happy to have all of you here with us. Just a few housekeeping things before we get started. As usual, I am going to try and mute everybody here on my end. Unfortunately, we are going to need you to make sure you mute yourselves as well, because both Kevin's supervisor, and Natalie, and himself have called in as a regular user, and so I can't mute you guys the same way I normally do without accidentally muting them. And so rather than having unexpected interruptions in the background, it's difficult for me to edit out before posting the presentations. We just ask that you mute yourself. Next week is Canada Day, so there will not be a session. So the next time we will be meeting will be July 8th. So just as an FYI, make sure none of you come to expect that there is something going on. So I am going to hand it over now to Natalie, who is Kevin Kuczynski's PhD supervisor, so she can actually introduce us to both Kevin, and then Kevin can go ahead and do his presentation. Thanks, Natalie.
Great, thank you. So it's with great pleasure that I introduce to you Kevin Kuczynski. He is a third-year PhD candidate, and he works at the BC CDC as well as at the University of British Columbia. So he's working on a project that Chelsea Hemsworth, William Zhao, and myself have been leading over the last 5 years or so in collaboration with the BC Ministry of Agriculture to understand alternative strategies for detecting avian influenza in the environment. And he's an absolute whiz at wet lab aspects of next generation sequencing, and has been very successful in getting a method up and going to be able to do some advanced testing of wetland sediments as a tool for avian influenza surveillance. And so with that, I turn it over to Kevin for his presentation.
Great, thanks for the intro. Can everybody see my screen here?
Yes, we can.
Okay, perfect, great. All right, well, I'll start off by telling you all a bit about why we care about avian influenza viruses. So they cause severe disease in poultry, and they're responsible for frequent outbreaks in commercial flocks. And we want to stop these outbreaks for 3 reasons. So first of all, they're extremely costly. The usual outbreak response is to euthanize the entire flock and impose a quarantine on all the nearby farms. So this usually leads to restrictions on poultry products in important export markets. So these outbreaks end up having a very big consequence for both the local and national economy. Now, the second reason we want to stop these outbreaks is because they expose farm workers to potentially deadly bird flu infections. Probably the most popular examples of these are H5N1 from the early 2000s and H7N9 a few years ago. These caused clusters of thousands of cases in China and Southeast Asia, with case fatality rates of around 30 to 60%, and both of these were associated with infected poultry. And this leads to the third reason that we want to stop these outbreaks, which is they create opportunities for novel flu viruses to adapt to humans. And this threatens future pandemics and eventually new types of seasonal flu. I think we're all pretty much now very intimate. We have a very intimate understanding of how disruptive a pandemic can be, and seasonal flu remains among the 10 leading causes of death globally. So preventing these bird flu outbreaks is a way to kind of nip all of these problems in the bud. Now, the ultimate source of all of our influenza problems are wild birds, especially waterfowl, so ducks, geese, swans, etc. They're natural hosts of influenza A viruses, and they're a vast reservoir of extensive flu virus diversity. And they cause these outbreaks by contaminating farmlands with their feces, which contains very large amounts of the virus. So consequently, most outbreak prevention and pandemic preparedness strategies involve surveillance of flu viruses in wild birds. Typically, this involves capturing live birds and testing them for flu, but as you might imagine, this almost never detects dangerous strains before they cause problems. So our team is developing a new approach that's based on targeted genomic sequencing of environmental specimens from the wild bird habitats. So as illustrated on this slide, we are focusing on the wetland sediment because this is where the virus-rich bird feces accumulates. We suspect that the sediment contains a pretty comprehensive collection of locally circulating flu strains, which makes it a really good surveillance specimen. It's also very easy to collect, and it doesn't require us to wrestle any angry geese and stick swabs inside the rear ends, so we really like it for that. Now, there is one big hurdle standing in the way of our whole mud sequencing scheme, and basically we're looking for needles in a massive haystack. So the wetland sediment is full of microbes and decaying plant tissue. So we extract a lot of very diverse nucleic acids from the sediment. But in contrast, the flu genomes we're trying to find in there are very small and they're usually fragmented. So the objective of my PhD research is to develop methods to capture, amplify, and sequence those rare scraps of influenza genome in the mud. I'll give you a brief overview of the method here that I've developed. It starts with extracting RNA from 2 grams of sediment, so that's roughly 1.5 teaspoons of mud. Then this RNA gets prepared into a sequencing library using a custom protocol that tries to minimize the loss of input material and retain those rare scraps of flu genome. Following the library construction, there are 2 rounds of enrichment using a custom panel of hybridization probes. These probes are synthetic DNA oligos. They're complementary to regions of the flu genome, and we use them to capture and pull down the genomic material we're interested in sequencing while we wash away a bunch of that background material. And then finally, following this enrichment, we sequence the libraries on an Illumina platform, and then we have some custom bioinformatics tools that we use to do the analysis. So our method currently targets 3 segments of the flu genome. The first 2 encode the surface proteins hemagglutinin and neuraminidase, or HA and NA for short. Since they're on the outside of the virus, they're under very strong pressure from the host immune system to mutate, so they're extremely diverse. And due to this diversity, they're traditionally used to subtype influenza viruses. So when you hear about H1N1 or H3N2 or H5N1, this is what those H's and N's mean. And we also target these segments because they also encode important determinants of virulence and tissue tropism and host range as well. Now, the third segment that we target right now is the matrix segment. It is the most conserved part of the flu genome, so it's a very reliable detection target for a diagnostic qPCR or for our hybridization probes. And we use it as a kind of control target that will show us that we've captured flu material even if we were missing other more diverse parts of the genome. So as I just mentioned, the influenza virus HA and NA proteins are very diverse and they evolve rapidly, which is part of the reason we need a new flu vaccine every year. Now, this diversity presented a really big challenge for designing the enrichment probes. So I had to write a custom software tool to analyze tens of thousands of flu genomes to find the most effective sequences to target with these probes. So for this project, the software generated a panel of 3,600 probes. And this plot here shows you how well those probes could detect these tens of thousands of example flu genomes. So each one of those little black dots is one flu genome sequence, and it's plotted on the x-axis according to how many of its nucleotide positions are covered by a probe. So, it's also stratified by genome segments, so the HA, NA, and matrix results are separated. So if you imagine the hypothetical perfect probe panel, all of these tens of thousands of little dots would be stacked up on the far right-hand side at 100%. But my goal was to design a panel where 90% of the sequences had at least 90% of their nucleotide positions covered. So the shaded orange boxes, those boxes contain 90% of the sequences. And you can see that for all three genome segments we're targeting, that bulk of the distribution is above that solid black line that indicates the 90% coverage mark. You can also see that the worst covered sequence out of all of these still had about 60% of its genome covered by these probes. So overall, it's not the hypothetical perfect probe panel, but we're pretty happy with the inclusivity of these probe designs here. So for the rest of the talk, I'm going to show you how well this method has worked on real mud specimens. So these results are from a preliminary batch of 23 sediment specimens. These were collected from 8 different wetlands around Vancouver between December 2016 and January 2018. And I've indicated the locations on this map here in case anyone's familiar with the Lower Mainland or Fraser Valley. So you can see it's a nice mixture of kind of, city parks, suburban areas, rural farmlands. Now, this slide here, what we did is we estimated the amount of flu material in all of these specimens by qPCR. So the CT values are all plotted, and you can see that they ranged from 31.3 to 38.8 with a median of 34.8. So if you know anything about CT values, this should be telling you that there's not a lot of virus in this mud. So I knew that these specimens were going to be a pretty good, challenging workout for my method. Now, this slide here shows you 2 important metrics from the sequencing run on these specimens. So on the left in blue, I've plotted the total number of sequencing reads generated, and then the percentage of those reads that were flu sequences. So you can see that about 82% of the sequencing data was flu, which shows you that the method really did remove a lot of that background material from the sediment. And then on the right in orange, I've plotted the number of libraries that contained some amount of flu sequence. So 22 out of the 23 libraries contain flu reads, which also shows that the method is quite sensitive, despite there not being too much flu in this sediment. Now, from a surveillance perspective, one of the most important questions is what strains of flu are present in these specimens. So on this slide, I plotted the number of specimens in which each of the HA and NA subtypes was found. So surprisingly, we found 9 HA and 9 NA subtypes, which we thought was a considerable amount of viral diversity to observe over the 14-month period that these specimens were collected. But for surveillance purposes, we also want to characterize these viruses beyond just their HA and NA subtypes. So for us, the most practical way to do that is to reconstruct their genomes using the fragments that we recover from the mud. So on the previous slide, you may have noticed that H11 was an especially common subtype in our sediment. So I selected one of those specimens and reconstructed an H11 genome segment from the fragments, the sequence fragments. Now, this plot here is showing you the kind of success of that reconstruction. It's showing you that 100% of the length of the HA segment was reconstructed, and that there is about 10,000 to 100,000 sequencing reads covering each nucleotide position. So in this particular case, it allowed us to reconstruct this HA segment with a fairly high degree of confidence. Now, one of the most useful analyses we can do with these reconstructed genome sequences is to put them on a phylogenetic tree with influenza viruses isolated from wild birds, poultry, and humans. So that allows us to see if the viruses we find in the mud are closely related to any dangerous strains that have previously caused trouble anywhere. So in this case, we can see that this particular H11, unsurprisingly, its close relatives they're all isolates from wild birds with no poultry or human isolates nearby. So this is, really reassuring from both an agricultural and public health perspective. You can also see that the close relatives of this H11 we reconstructed are all isolates from North American wild birds from recent years. So for us, that was a really nice sanity check to show that this method generates the kind of results that make sense, given when and where we collected these specimens. So going forward, we are hoping to use this kind of data to better understand flu migration. And this could help us collaborate with other jurisdictions on migratory routes to build bigger regional or even continental surveillance networks. And then going forward, we would also like to use this kind of data to look for strains that contain genotypic markers associated with high pathogenicity in poultry or the capacity to replicate in livestock or humans. So all in all, it's a lot of questions to be able to answer just out of a couple of teaspoons of mud. So to wrap this up, these results showed us that it is technically possible to recover and reconstruct influenza genomes from the sediment using this custom method. So, the technology is there to do it. There are, a lot of, operational considerations, though, about how to actually deploy this. So right now what I'm doing is I'm scaling this up and applying it to around 230 sediment specimens so that we can get a big data set. And we're going to work with our collaborators at the BC Ministry of Agriculture to look at those data and then figure out a way to actually deploy this technology in upcoming years to do bird surveillance in our province. Now, I'd also like to mention that we've tried to make all of these methods easy to repurpose. So basically, we want to make it easy for other groups to do this kind of thing, even if you're not really interested in bird flu and mud. So, there's all kinds of applications you can imagine where you have a very diverse genome in a very difficult specimen where you might want to do this thing. So we're going to be publishing the probe design software as well as all the laboratory protocols for making the libraries and doing the probe capture. And hopefully those will be out later this year, depending on, the research situation that continues to evolve with COVID. So with that, I'd like to thank the INKY group for putting this together and inviting me to speak. I think these kind of One Health venues are really important. And of course, I'd like to thank my supervisors and other members of the Avian Flu Project for all their input, and also our funders for all their generous support. Thanks a lot.
Great. Thank you, Kevin. That was incredibly interesting. We do have a fair number of questions that are coming up. So what we'll do for now is if you just submit your question to either Andrea or myself via the chat, I will ask them of Kevin. If we end up having a bit of a slowdown, we could potentially open it up a little bit later for a more fulsome conversation. So the first question, Kevin, is given how incredibly useful this would be in a variety of different situations with environmentally survivable pathogens, so not only avian influenza, Seneca Valley virus would also be a good example of this. Do you think that this is something that could be used in a variety of settings other than just with mud? Or is it the thing that it's much easier to do it if you can get the fomites and stuff like that off, like within that kind of a texture?
Yeah, that's a deceptively complicated question there. So that really depends a lot on the kind of specimen matrix you're dealing with. And so one part of the story that I didn't really get a chance to talk about was the nucleic acid extraction part. And different types of specimen matrix call for different types of extraction methods. So once you get the RNA or the DNA, as the case may be, these methods that we have here are pretty modular, and you can kind of just slot them in. So I would say that probably the most complicated part of repurposing this is going to be figuring out the best extraction method to use on your specimens. And, that's going to really depend on what you're trying to do. And it'll probably require you to do a bit of experimentation to compare what kind of yields and recovery you get using different methods.
And extraction is always the big issue.
Yeah, it's also the big throughput bottleneck as well.
Right, okay.
So you got to find a way to get that method that balances recovery with also not being so painfully tedious that nobody wants to do it.
Yeah, exactly. Okay. So then next question, did the sediment sampling target any specific time of year? So knowing there are, especially for waterfowl, there are very definite migratory patterns. Was there a specific time of year that you would be looking at?
Yeah, so actually, so I'm one of a couple of PhD students on this project. So we have a veterinary epidemiology student named Michelle Coombe, and her project is, she's the one, first of all, who had to suffer out at these wetlands collecting mud at all times of year, surrounded by angry birds. So her project is asking those kinds of questions, like where and when do you sample? And I don't want to spoil her punchlines too much, but yeah, there's definitely a seasonality to it. And that's partially going to be due to the migratory patterns of the hosts, of the wild birds. But then there's also the environmental considerations. So, the temperature of the water and the pH of the water will change at different points of the year, depending on how much rainfall there is, for instance. And that chemistry will affect the recovery of the virus from the sediment. So, there's a combination of factors that all conspire to make certain months definitely better than others for doing this.
I know Michelle quite well. I'm guessing she was actually in her element doing that collection.
Oh, yeah. I went out with her to do some sampling, and it was a lot of fun. Yeah.
Okay. And so then, has the method been used around farms at all during an outbreak? Because it would be interesting to know how the virus survives depending on the actual viral load. Right.
So there was a big outbreak here in 2015 in H5N2, and this kind of technology, that's kind of what, that's where like the kernel of this project came from, was that outbreak. So they were able to detect the outbreak strain in wetlands around the affected farms using qPCR specific to that outbreak strain. So that's what really gave them the idea that, maybe we should just go looking in the mud and try to find the viruses there so that we don't have to bother the birds. So I wouldn't exactly say I'm hoping for a bird flu outbreak, but, if one does happen or when the next one does happen, it would be interesting to see, how widely detectable that strain is in our Fraser Valley region, and how soon before and how long after the outbreak we can detect it as well.
Well, speaking as an epidemiologist, the reality is that things are always better when you have an outbreak, right? Because you actually have the ability to have numbers, whereas Otherwise, everything is somewhat untested.
Yeah.
So, particularly in the case of migratory waterfowl, would you expect to see closely related influenza genomes or subtypes in winter ranges of these species?
So, yeah, I think that really highlights in the zoonotic influenza field. I mean, at this point, humanity has a pretty good grasp on in human, like seasonal influenzas, but I don't think that we know too much about influenza diversity and dynamics in wildlife. So it would be interesting to see, you know, if it's kind of like us, where different species of birds have kind of endemic subtypes of flu that are stable for decades and then change abruptly when they get a spillover from another species of wild birds, and then to see if, you know, they're carrying their subtypes of flu around the world with them throughout the year, or if it kind of changes depending on where they are on the planet. So this is where it'd be nice to have more people doing flu surveillance at an environmental level in different jurisdictions, so that we can try to put those pieces together. Because right now we're kind of just getting little snapshots in space and time that are very specific to, you know, one scenario.
Yeah, like with anything really where you're studying wildlife.
Yeah.
So can you comment then on the significance of finding nucleic acid fragments versus testing actual bird specimens or samples?
How do you mean the significance of it?
Keep in mind these questions are coming to me from other people.
Oh, okay.
I'm guessing whether or not it's more of a confidence rather than significance. So if you are talking about statistically, there's confidence and uncertainty, right? Always in what you're calculating.
So I don't think that this replaces bird surveillance. I think this complements it, because there is a lot of uncertainty in this method. When you're finding the scraps of genome, we don't know when that virus was deposited. You know, there's also this question of, you know, like, have we developed a method that's too sensitive? You know, like what do you do if I find one single scrap of highly pathogenic H5? Do you have to report it to the OIE? Do you got to shut down the farms? Do you do nothing? Do you dismiss it as an artifact? You know, so there's a lot of those kind of questions about how to use this technology practically that still are unanswered. So I think probably what we're imagining is that this would complement bird surveillance. So we would use this kind of as a way to do an extensive screening surveillance of the local area, and then if this genomic environmental surveillance turns up something troublesome, like something concerning, then the reaction would probably be to increase bird surveillance to try to isolate that virus and really confirm that it's around and it is the dangerous one that we think it is.
Okay. So similarly to the difference between PCR, which just says, yes, it's there, versus virus isolation, which tells us that it's actually active at the time of blood sampling. Could be used on a more regular random basis to sample a variety of different environments, and then when you find something troubling, then you could sort of focus in and do more targeted active surveillance on a number of farms in the area.
Yeah, exactly. Yeah, I mean, both methods have strengths and weaknesses, and you're going to have to put them together, I think, to really deliver the best surveillance
Perfect. Okay, so one last question then. If you had been hoping for someone to ask you something specific or something more detailed about your project, what would you have liked it to be? And go ahead and answer that question.
Oh boy, that's I think probably one question that I'm kind of surprised that we didn't get, and the one that comes up often, is how stable in the environment are influenza viruses. And yeah, that's a very controversial topic, surprisingly, in the flu field. I think, so I mean, there's some people that don't think that they're going to be stable at all, and that the only thing that we're ever finding here are, scraps of, virus that are not viable and nothing to worry about. But there's been some interesting work where people have, there are people who do the bird surveillance, and, occasionally they will find a genetically identical virus, but separated by an entire season. And given how quickly influenza evolves and mutates, that's just not really practical to assume that, you would isolate a genetically identical virus months apart like that. So the explanation has been that some have reached, and that, obviously we're going to be partial to, is that this is just evidence that there is some environmental reservoir of flu viruses where the viruses get deposited there, they go into stasis, they remain viable, and then at some point later, especially after a winter, for instance, where the temperature drops, so it's going to be better preserved, that virus gets reintroduced into the birds. And obviously, we'd like to believe that environmental reservoir is the sediment. Now, the sediment, what's interesting is that it's composed of, little crystalline mineral particulate that has, really strong charged surfaces on it. And then those viral proteins, depending on the pH of the water, they also have a charge to them, and an electrostatic charge. So they end up, coating the surfaces of these mineral particles and sticking to them, and then the mineral particles clump up, and it's thought to actually form this kind of protective cocoon for the viruses that, helps them survive in the environment for extended periods of time. So I think we're kind of becoming more and more converted to the camp that influenza viruses are going to be persistent in the environment for quite some time. And, when you think about it, it makes sense that they would be. Because, I think we have this perspective of flu viruses as a fragile respiratory pathogen because they don't survive very long on doorknobs or elevator buttons or whatever. But fundamentally, influenza viruses are a waterborne infection of birds. So we probably have to give them some credit for their ability to survive in an aquatic environment.
Good point. Okay, so Kevin, we're at time. And so in the guise of making sure that everybody can go back to work, we're going to end things now. But thank you so much for presenting.
Thanks a lot for having me.
Really interesting. And I might actually follow up with Michelle and see if she can speak to us in the fall on her work as well, because it would be interesting.
Well, I'm sure she'd love to.
Oh, yeah. And if you want to talk to her as well, if you're in contact before I manage to do so, that would be also great. If anybody has any further questions for Kevin, please don't hesitate to send them to me, and I will forward them on to Kevin and have him answer, and then we will add them the next time we send out an email. Just a reminder that next week is Canada Day, so everybody who is going to be celebrating that day, it doesn't always get celebrated by everyone, but everybody who's going to be celebrating, please enjoy yourself, but do remember to stay safe and wash your hands. Bye, everyone.
Thanks again. Bye.